So what is DNA replication in the sense? From the parent DNA, you are finding two daughter DNAs are coming. That is the meaning of DNA replication. Okay. So recombination and repair also we will see as we proceed. Okay. Now basically in this chapter like we have two topics like the DNA replication which is seen in the prokaryotes and also in eukaryotes. So prokaryotes we need not go into detail, whereas eukaryotes DNA replication usually comes as an essay question. Okay. So the central dogma of life, what does that mean? That means from the parent DNA, you will see daughter DNA is formed. From the daughter DNA, you will see like one of the strands of the DNA will be read and it will be moving in the form of messenger RNA from the nucleus into the cytoplasm. That mechanism is transcription. Messenger RNA will be carrying all information which is required for the synthesis of protein, that is translation. Okay, that's what in this long lengthy chapter of molecular biology, one after the other, we are going to learn. Transcription, first replication, then transcription, then translation. So finally, you will see the formation of protein, because most of our body building things are nothing but proteins only. Enzymes are proteins, hormones are proteins, muscle is a protein, bone is a protein, isn't it? Most of the clotting factors are proteins. So all these are synthesized. Like in this chapter, we are going to learn the details of this. Okay, so that is central dogma of life. Now, coming to replication of DNA. So suppose this is parent DNA. So parent DNA, two different colors have been used here. One is green and the other is blue. Okay. So the parent DNA strand will uncoil itself. Now one side you can see green color has come and the other side you are seeing blue color has come. Okay. So usually on the parent DNA, you will find strands like we have adenine, guanine, thymine, cytosine. These are the nucleic acid which we see, which is carried by the parent DNA strand. So there is nucleotide base pairing, isn't it? So like we always know, like adenine pairs with thymine, ATGC, guanine pairs with cytosine. Between AT, you will see triple bond is there, and between GC, we have double bond. So one of the parent DNA will carry all the information of the required nucleic acids. So correspondingly, the base pairing starts and the daughter DNA strand is synthesized. So you can see from one parent DNA, two daughter DNAs are formed. So semi-conservative in the sense, half of the identity is coming from the parent DNA and the remaining half is synthesized based on the information which is brought by the parent DNA. That's what is called as semi-conservative mechanism, what we see in DNA replication. Okay, so replication in prokaryotes. Prokaryotes are nothing but the bacterial and primitive cells. Okay, so replication in prokaryotes generally it will be in a different way, in a much simpler way, which we are not going to see the details. But this word Okazaki pieces is actually important, which is also applicable for replication in eukaryotes. Now, what are these Okazaki pieces? Let us see first and then come back here. Now, suppose this is parent DNA strand, which is shown in 3', 5' direction. Okay. Then newly synthesized DNA is seen like associated with the parent strand. One side it is shown in red color. This is the newly synthesized DNA. So you will see the newly synthesized DNA will be coming in the form of pieces so that it will be fast. The replication mechanism is fast. Okay, the base pairing will be very fast and the daughter DNA is formed in a faster pace. So for which you will have pieces of strands of mini DNAs, which is nothing but called as Okazaki pieces. Okay. Now you can see this is one Okazaki piece. Okay, like it is written Okazaki piece. This is one more Okazaki piece. So which will have required like nucleotide base pairing elements. That is nothing but Okazaki piece. Now let us read. See here, the small fragment of discontinuously synthesized DNA are called Okazaki pieces. Discontinuously synthesized, that means this is one piece, this is one more piece. So if you find such discontinuously synthesized DNA strand pieces, so these are nothing but Okazaki pieces. These are produced on lagging strand of parent DNA. Lagging strand is this beta. Leading is the one which is coming from the parent, and the one which is newly synthesized is nothing but the lagging strand. So here you are finding pieces. Okay. So Okazaki pieces are later joined to form continuous strand. That means whatever pieces you are finding now, this will be ligase enzyme will be coming, and it will be just sticking these two ends. Okay. That is later joined to form continuous strand of DNA. So DNA polymerase 1 and DNA ligase are responsible for the process, which we are going to discuss in detail now when we take the replication in eukaryotes. Okay. We will see replication in eukaryotes. Very, very important. It comes as an essay question. Okay. Now see here. Replication of DNA in eukaryotes. First thing is I will try to show you everything from this picture, then you will learn. Now before that we will try to know about few enzymes. So what are the various enzymes? See, replication of DNA in eukaryotes closely resembles that of prokaryotes. A few like important informations like Okazaki pieces and all I have shown you in prokaryotes. So, but certain differences will exist. So basically for the formation of daughter DNA in case of eukaryotes means advanced cells like human beings and all. So in advanced cells, whatever mechanism of replication is taking place, now we are learning that is replication in eukaryotes. This is only will be asked in your exam also. So we require 5 different DNA polymerases for the synthesis of daughter DNA. Okay. So what are those DNA polymerases? We have DNA polymerase alpha, beta, gamma, delta, and epsilon. Okay. Now coming to DNA polymerase alpha, what does it do? See, it is responsible for the synthesis of RNA primer. RNA primer is nothing but same like Okazaki piece what we have seen in case of prokaryotic. Okay, RNA primer for both leading and lagging strands. I will show you in detail as we proceed, but few words which you should understand here. So DNA polymerase alpha, then we have DNA polymerase beta. It is involved in the repair of DNA. Okay. So suppose if something goes wrong, so beta is helping to repair that. So its function is comparable with DNA polymerase T, which is found in prokaryotes. Then we have DNA polymerase gamma, which is participating in the replication of mitochondrial DNA. Okay, and so delta is responsible for the replication on leading strand of DNA. It is also possessing proofreading activity. See, as we see, you will learn more better. And this epsilon is involved in DNA synthesis of lagging strand and also proofreading function. Now, process of replication in eukaryotes. Let us see. Now see here, you are finding, see, in case of replication, first thing what happens is uncoiling of the parent strand. Okay, only one strand from the parent is coming, isn't it? That's what I told you, semi-conservative mechanism. So this strand has come from the parent. So this is 3', 5' direction you are seeing here. Okay. Then here in the other side, you will see all base pairing will start. So if we just find at the 5' end of the parent DNA template, this is the template for lagging strand. You are finding Okazaki pieces have started coming. So previous Okazaki fragment is here. Now, here you are finding something like RPA. So what is this RPA? So the RPA is nothing but the replication protein A. As we proceed in this mechanism of replication, you will see the replication protein A's will be removed. All these small balls will be removed. Beta, you can see. You see overall, if you see in the last, uh, like uh, uh, like last step, you are not finding even single replication protein A. So when the base pairing mechanism is taking place, gradually the replication protein A's will be removed. Okay, and Okazaki fragments will be getting added from both the directions, from 3' and also 5' direction. They will be working in two directions. Now in the first step, what we are finding, RNA primer. This is also same, more or less like Okazaki piece only, which is helping in base pairing. And the RNA primer has started coming from both the directions, 3' and 5', isn't it? So RNA primer is now brought by the polymerases. Just now I have told you 5 different types of polymerases, right? So DNA polymerase alpha primase complex. Okay, beta. DNA alpha polymerase primase complex has brought this RNA primer. So RNA primer has come from 3' direction, also from 5' direction. And these replication protein A's are being removed. Now this cycle is moving actively in both the directions. Okay. Then you are finding PCNA and replication factor C. They have come up to speed up the process. Proliferating cell nuclear antigen and replication factor C will help in faster movement of these RNA primers, so that base pairing is occurring fast. Okay, Shubhangi, understanding?
Yes, ma'am.
So see here, now the DNA polymerase delta also has come, and the polymerase factor, replication factor C, they are moving faster and these proteins are being removed. Okay, only 3 proteins are left over, RNase, and now you see this flap endonuclease 1 also has come. Gradually, the RNA primers have moved from both the directions, 3', 5' directions, and finally they are almost very much close to come and get glued, for which we require an enzyme, that is DNA ligase. So the DNA ligase has helped in binding up the patch. Okay, so the new strand of DNA is finally prepared. Okay, beta. So this is
Yes, ma'am.
how the daughter DNA strand is prepared in response to the information which is brought from the parent DNA. Base pairing mechanism takes place, nothing but. Okay, so you have to give the list of like endonucleases which are actively working, the 5 enzymes like polymerases, DNA polymerase alpha, beta, gamma, delta, epsilon. Okay, and then you have to give all these steps, and whatever you see in the diagram, same thing is written in the form of text. Just practice that flowchart and just write these points, you will get full marks. Is it clear?
Yes, ma'am. Yes, ma'am.
Then we have inhibitors of DNA replication. So that means whenever we don't want replication to happen, suppose if cancer chapter we have studied, unnecessarily we are finding cells are multiplying. You don't want cells to multiply. Then you will give certain anti-cancer drugs, if you remember. There also we have studied,
Yes, ma'am.
right? 6 mercaptopurine, 5 fluorouracil. So we have certain agents which can stop replication. So which are nothing but inhibitors of DNA replication. Okay, madam?
Yes, ma'am.
Cell cycle is not required. That is nothing but precursor of mitosis and meiosis. Telomers and telomerases also you don't require. We don't even require these recombinations. It has never been asked. All these are not required. Okay.
Yes, ma'am.
Just because you have time, you can just read this table better. What are the types of DNA damages? Okay, single base alteration, cytosine with uracil instead of adenine, hypoxanthine, depurination. Just go through with this table and try to learn this. Check also with question bank if such questions were asked. Mutations are very important. Sudden change in the genetic information, genetic makeup is nothing but mutation. Change in the DNA structure of a gene. Okay, and the substances which can cause mutations are called as mutagens. Okay, certain chemicals are there like UV rays, gamma rays, X-rays. Suddenly they can change the genetic information of DNA. So obviously the protein which will be synthesized also will change. So those agents which can change the genetic information, they are called as mutagens. Okay. So we have different types of mutations, beta, like point mutation. What does it mean? Replacement of one base pair by another. That means instead of 80, something GC may come. Okay. So which is called as point mutation. Okay. So under this we have two types. Transition is there, transversion is there. In case if purine is replaced by like purine or pyrimidine is replaced by another purine or pyrimidine. Purine for purine, pyrimidine for pyrimidine only. You call it as transition. Okay. Transversion is instead of purine you are finding pyrimidine is coming and vice versa. You call such a change in the genetic code, you call it as transversion. Next, we have frame shift mutation. What does that mean? Frame shift in the sense, like a particular part of the genetic information is being shifted. Like ATGC, one code word is there, genetic code. 3 or 4 codons are being cut and they are being pasted at the end of the genetic, that information, 3 prime, 5 prime end. Big things are being cut and being pasted. So that is frame shift. These occur when one or more base pairs are inserted or deleted from DNA. It may be causing insertion or deletion mutation. What are the consequences because of mutations? First of all, we are seeing with point mutation. So point mutations are single. It can be transition or transversion, right? Only one particular purine or pyrimidine is being replaced with same purine or a different pyrimidine, right? So in such case, we know, like if you remember, Google hypothesis. In case of Google hypothesis, we will learn like 3 or 4 codons are coding for only one amino acid. Isn't it?
Yes, ma'am.
So suppose if UCA is coding for suppose serine, UCU is also for coding for serine only, such mutations will not cause any harm to human beings. So such mutations are called as silent mutations. Whether it is UCA or whether it is UCU, A has been replaced for U. Both are coding for serine only. So such mutations will not cause any harm. And such mutations are called silent mutations. Okay, beta?
Yes, ma'am.
Then we have missense mutations. So in this case, changed base pair may code for a different amino acid. Like UCA is basically coding for serine, but now U has been replaced with A, which is coding for threonine. So this is, of course, not actually, it is also little harmful only. So that is missense mutation. But it may be acceptable or partially acceptable or unacceptable based on what protein is synthesized. Like sickle cell anemia, if you remember, there also only one amino acid is replaced, no? Glutamate is replaced with valine.
Valine.
So such small change only, but it will cause change in the shape of RBC. Instead of biconcave disc, they become sickle shape. So that is also very harmful. So it can be either acceptable or partially acceptable or unacceptable, as you can see in sickle cell anemia. Okay, beta?
Yes, ma'am.
Then we have nonsense mutation. Sometimes the codon with the altered base may become termination codon. That means instead of UCA, if C is replaced with A, the chain length of protein will not elongate only, because UAA will not code for any amino acid. That is stop signal, stop codon. Okay. So that will stop the further synthesis of amino acid insertion in protein chain. Do you understand?
Yes, yes, ma'am.
Stop signal has come, so the elongation of protein chain will not happen. So that will not cause any harm. That's the reason we call it as nonsense mutation. Okay.
Yes, ma'am.
So frame shift mutations are of course very dangerous only because large, very big genetic codons, no, 3 to 5 to 6 codons are cut from one place and they are pasted to some other place. So obviously, a different type of protein will be synthesized, which is a novel protein and which may be not useful to the body. Okay. So in case of frame shift mutation. Okay. So mutations also can cause cancer, what we have studied in cancer chapter, isn't it? So now repair of DNA. So this now better you read from this table better. Base excision repair. Damage to single base due to spontaneous alteration or by chemical or radiation means removal of base by N-glucosinolase. This is the repairing enzyme. This is the damage caused. Just read this table. Only table is enough, better. Don't read all these things, not required.
Yes, ma'am.
That finishes this chapter better. Okay. So plasma proteins, basically in the plasma, whatever proteins are there, like what are those? Albumin, globulin,
Globulin.
fibrinogen, all these are called as plasma proteins. Now, where do we find plasma? In the blood, isn't it?
Yes, ma'am.
What we call serum. So serum has
Serum.
proteins. Along with proteins, we use the word plasma proteins. So what is the basic importance of plasma proteins? What you should know is plasma proteins are very important for the maintenance of osmotic pressure, also called as oncotic pressure. Okay. So which will not let the fluid component of blood to leak into tissue spaces. That means these plasma proteins are very important to retain water in the blood circulation. If plasma proteins are not there, the water content of the blood will leak into tissue spaces causing edema. The blood volume should be at least 5 liters in healthy 65 kg adult. But in case due to protein deficiency in the diet or due to protein loss due to some diseases, what we will see, like in case of diarrhea and dysentery and all. So what happens if proteins are lost from the blood, then person will suffer with retention of water in the body. That is nothing but edema. Okay, that is the chief importance of proteins, plasma proteins. Okay, beta? That concept you should know to understand many subjects as you go in this course. Okay, in many subjects. Electrophoresis. Yeah, electrophoresis can be asked, beta. So plasma proteins, we know the names. Commonly employed analytical technique for the separation of plasma proteins. We use electrophoresis to separate, see, plasma proteins like albumin. Globulins are alpha 1, alpha 2, beta, and gamma. Okay, so these are globulins. So electrophoresis of plasma proteins. So these are the separated bands. Okay, this is one more mechanism which is called as densitometer scanning. Densitometer scanning is not so important, but usually this electrophoretic technique which will help in the formation of separate bands, which will help us to identify whether it is albumin or fragments of globulin. Okay. Now, abnormal electrophoretic proteins we generally find with certain diseases. Multiple myeloma, it's bone marrow cancer, better.
Yes,
Acute
ma'am.
infections. Nephrotic syndrome, in that case, you will see heavy loss of proteins in the urine. You call that condition as nephrotic syndrome. Okay. Immunodeficiency disorders like HIV, AIDS, better. There also you will see gamma globulin levels are decreased. Antitrypsin deficiency also. Okay. In this chapter, no, Shubhangi, what you should remember is this one better. This table you try to memorize. That too, not everything. I'll tell you what to study. See, here we are given the list of the names of proteins, and here we are given the functions. So functions of plasma proteins is usual question. So here what you will write is, suppose if it is albumin, its inactive form is called as pre-albumin. Remember the plasma concentration of albumin because it itself can come as a 3 marks question. 3 to 5 grams per deciliter. Don't remember molecular weight. Function, osmotic transport. Then osmotic regulation of osmotic pressure helps in the transport of things like what are those hormones, growth hormone and all. Then nutritive function also, and it also acts as a buffer. We have studied, no, protein buffer in
Yes,
blood.
ma'am.
So pre-albumin also helps in transport of certain hormones like thyroxine to some extent. So albumin you remember better. Globulin you just give the names of the subcomponents. You need not remember plasma concentration, or only one you can remember, alpha 1. You need not remember molecular weight at all. Just read the functions. Read the functions of everything now. Just give the names and read the functions. That is more than enough. Okay. Okay, beta. This is important. Ceruloplasmin. This may come as a 3 marks question. Before that, let me check anything if I missed something. Now, ceruloplasmin. If you remember, when we were doing like iron metabolism.
Iron, ma'am.
Very good. So there copper, functions of copper also we have come across this, right? So blue colored copper containing alpha 2 globulin with a molecular weight of around 1,50,000. So in the plasma, its concentration is 30 milligrams per deciliter. So ceruloplasmin binds with almost 90% of plasma copper. Okay. So it is very tight and it is not easily released in tissues. Okay. Albumin is carrying only 10% of copper, whereas ceruloplasmin carries 90% of copper in the tissues. So it helps in oxidation activity also. Biological oxidation. In that chapter also we are going to see this ceruloplasmin. So suppose if it is deficient, then you will see this Wilson's disease. Did we discuss this in copper metabolism?
Copper, yes, ma'am. Menkes disease and Wilson's disease.
Yeah, very good. Excess copper is
Wilson's.
Okay. So transferrin. So this is also sometimes it may be asked like it is associated with beta globulin. It helps in the transportation of iron, isn't it?
Yes, ma'am.
Acute phase proteins, we will see these are something new proteins which are seen in new diseases are coming up. No, nowadays, like COVID itself is a new disease, right? For 2020. So like that, we have many viral infections during which new proteins may be seen in the blood, which are called as acute phase proteins. So due to infection, injury, or due to some inflammation, we will see some new proteins are seen in the blood, which are called as acute phase proteins. Okay. So alpha-1 antitrypsin, ceruloplasmin. So some different shape and structure they will be possessing. C-reactive protein. It is produced in the liver and it is found like in a very small concentration. Like this is seen normally in complement cascade. Complement is also one of the like you have clotting factors like many in 13 in number. Similarly, see we also have this C-reactive protein as component of complement, which you will be studying in second year microbiology in detail. It actually helps in active working of antigen antibody complexes. To kill the antigen, complement will be assisting. So in that complement cascade, you will see the C-reactive protein will be working. So generally, if a patient is undertaking any surgery, such proteins will increase in blood. Immunoglobulins are nothing but antibodies. Try to learn this structure better. Very, very important. There is
Yes,
a
ma'am.
code for immunoglobulins, GAMD. Again, this table is so good. GAMED, GAMD, immunoglobulins. So heavy chains and light chains. Which are heavy chains? See, this side, whatever we have, better this one. This is heavy chain, this is heavy chain. The small strands are light chains. Okay, these are light chains. L chain is light chain, H chain is heavy chain. So names are given. Suppose if it is immunoglobulin G, heavy chain is named as gamma. Light chains can be either kappa or lambda. Molecular formula is gamma 2, kappa 2 or gamma 2, lambda 2. Molecular weight is 1,50,000. So percentage of carbohydrate in this basically they are proteins, no? But 3% of carbohydrate is also there in the structure of immunoglobulin G. Serum concentration is 800 to 1500 milligrams per deciliter. Functions responsible for humoral immunity. Okay, beta? So like
Yes,
that,
ma'am.
you please read this whole table. Whatever immunoglobulin is being asked, you can elaborate from this. Okay. Benzones proteins may be sometimes seen in the urine beta. Very small sized proteins which are sometimes filtered in the glomerulus. In early stages of renal disorders, we call such proteins as benzones proteins. Very rare question. Amyloidosis is also important essay in pathology second year. Now you will not even understand it. It's a 1 hour lecture in fact. Blood clotting is simple what you study in physiology, but not important in biochemistry. Anticoagulants also, like you just try to remember the names like oxalate, fluoride, ADTA, citrate. Don't read this at all. This is for physiology only, but given in biochemistry also. Okay, so that finishes plasma proteins as well.
Yes, ma'am.